Hello, students. A very good afternoon. I welcome you all to the today's session where we are going to have a very quick revision on biomolecules. So in this session today, we are going to be concentrating on very important topics. So recently when we had a talk on biomolecules, so basically I will go with an introduction for a few minutes. So biomolecules are essentially some of the chemical compounds that are very, very important for the entire construction of living organisms. So we have heard biomolecules in different levels of our academics. It might be sugars, it might be lipids, it might be polysaccharides, it might be nucleic acids, etc. So before we start on to today's topic, so I would like to go with the first question. Okay, so here we have the first question. Before we start the topic, so I'll give you 1 minute to solve the question. So here we have the two functional groups characteristic of sugars are. Okay, so I'll launch the poll. And before we have this question, can I have a technical check where you can see my video as well as hear my voice properly? Can I have a quick reaction on it? Yes, thank you. So anyways, I hope that you did not listen to the first first introduction session. I was not talking much, but I was telling that we are going to discuss a biomolecules chapter where we'll have a quick revision on it, if that's all right. Okay, so before we start the topic, we have a question here, and the question is, the two functional groups characteristic to sugars are, okay, then we have hydroxyl and methyl, carboxyl and methyl, carbonyl and phosphate, carbonyl and hydroxyl. So I'll launch the poll. Try to answer the question. Okay, carbonyl and phosphate is the answer that is provided. Anyways, I'll wait for another 10 seconds. Okay. So anyways, going with your answer, actually the two functional group or two functional characteristics of sugar groups are, see, if you see this picture, you can clearly understand this. So first we have a hydroxyl group, okay, which is an OH group that is attached to the sugar molecule. And then we have a carbonyl group. So our option for this question would be carbonyl and hydroxyl groups are the two functional groups of our sugars. Okay, so I hope you can see my ink. If you have a problem in the ink, I will change it to black color or something like that. Okay, I'll change it to black. Okay, for now it's visible, I feel. Anyways, so we have a second question here. The major role of minor elements inside living organism is to act as. The first option we have is cofactors of enzymes, building blocks of important amino acids, okay, constituent of hormones, okay, binder of cell structure. So I'll launch the poll again. 1 second. Yes. So here is your poll. You can answer this question. Okay, if you can see, I hope you can see it. Yeah. The major role of minor elements inside living organism is to. Exactly. So you have given the answer in less than 35 seconds, which is very fascinating. Okay, and your answer is absolutely right. Okay, the major role of these minor elements, which are usually some of the ions, okay, they act as cofactors for the enzyme. So your option is very much right. So coming to today's session, so we have to cover some of the very important topics with respect to carbohydrates, proteins, lipids, and enzymes. So let's get on with today's session and cover each topic. Okay. So first, initially, we will talk about biomicromolecules. So when we are talking about living organisms, so we talk about how living organisms are made up of cells, tissues, organs, and organ systems. But when you look at the molecular level, we are all made up of atoms and molecules. So these atoms and molecules, they combine in various forms to provide us with biomicromolecules or biomacromolecules. So the basic way to understand what are the ingredients that we have in our body, we take a living tissue like a liver or a plant cell, and then we will treat it with trichloroacetic acid. So we treat it with like trichloroacetic acid is because the tissues are very well attached, right? The cells are highly attached to each other, so that it will help them to free themselves. So once we grind them into a paste and then filter them, so we will get a filtrate where we will have two different pools, right? One is acid, okay, acid soluble pool, and then acid insoluble, right? Acid insoluble pool. So basically acid soluble pool will have all our biomicromolecules, and acid insoluble pool will have our biomacromolecules, and this is how we study them. And then if you want to go like elemental state, what are the inorganic or organic compounds that are present in our living tissues? So basically what we do is we take these tissues and burn them. So when we burn them, so we get ash. So in this ash, if we try to run a test, we will get all this organic and inorganic compounds like sulfur, oxygen, nitrogen, phosphor, okay, calcium, etc. So that gives us a very clear idea about what our living body is made up of. Basically, our living body is made up of the same earth-like elements, like you have your both organic and inorganic elements. So coming to biomicromolecules or macromolecules, so these are highly important to us, okay, in building some of the important structures of our body. I mean, like we will go with the cells, okay, some of the important structures of our cell, be it amino acids or lipids or sugars or nitrogen bases. So they all belong to a category of biomicromolecules. And why do we call them biomicromolecules is because of their molecular weight. So if we have any molecule that is ranging between 18 to 800 Daltons, so then we call it as basically biomicromolecules. Okay. So where do we find this biomicromolecules? So we can find majority of the biomicromolecules in our cytoplasmic composition. Okay. Our cells have cytoplasm, right? So within the cytoplasmic composition, we have all these micromolecules. So we will study what is amino acids and what are lipids, sugar, and nitrogen bases in detail in the coming session. Okay. So coming to biomacromolecules are basically present in acid insoluble pool after we extract it. So their molecular weight is greater than 1000 Daltons. Okay, they are mega molecules basically. So these molecules are very fundamental for the entire construction of a living system. So they include your proteins, okay, polysaccharides, which give you energy and storage of energy, and nucleic acid, which basically store all your genetic information. Okay, so this is about biomacromolecules. So let us study one by one. So initially we will start our session by discussing about carbohydrates. So carbohydrates are usually carbon, hydrogen, and oxygen containing compounds. Okay, so these carbohydrates can exist in two forms when it comes to geometry or the you call it as molecular structure. One, it can be normal like a chain, or it can have a ring structure to it. So usually the chain structures are in solid forms, okay, and the ring structures you can find in when it is dissolved in water, right? So in aqueous solutions, you can find this ring-like structures. So sugar molecules can have a variety of ranges. For example, it can be simple sugars, for example, glucose and ribose. Okay, and then it can be of complex sugars where you have disaccharides and you have polysaccharides. So based on the characteristics of the sugar, right, based on whether it is a single sugar molecule or it is more than one sugar molecule, we can categorize them into following categories. So carbohydrates can be divided into monosaccharides, disaccharides, oligosaccharides, and polysaccharides. So basically, when it comes to disaccharides, oligosaccharides are the part of disaccharides, I can tell you. So when it comes to monosaccharide basically contains of one single sugar molecule, that is only one type of sugar molecule. So if you take glucose, it involves a repetition of 4 to 8 repetitions of the sugar molecule. For example, if I say glucose, that is C6H12O6, it will be repeated like how many times? It will be going up to 4 to 8 times. Okay. For example, C6H12O6 for another 5 times, something like a chain. All right. So this is about monosaccharides. So examples of monosaccharides are glucose, fructose, and galactose. Okay. So when it comes to disaccharides, it involves 2 or more sugars. Okay. Basically it involves 2 sugars which are having the repetition of 2 to 8 repetitions. Okay. For example, sucrose, lactose, and maltose. Okay. So if I take sucrose contains glucose and fructose. Okay. It contains what? Glucose and fructose. Okay. If I take lactose contains, what does it contain? Glucose and galactose. Okay. Similar to that. Okay. It is made up of 2 different sugars, isn't it? Glucose, galactose. Here you have glucose and fructose. Okay. So this is about disaccharides. When you take about oligosaccharides are similar to that of your disaccharides, but the number of repetitions is quite high compared to that of the disaccharides. So this is about oligosaccharides. Okay. So when it comes to polysaccharides are complex sugars. Either it can be homopolysaccharides or it can be heteropolysaccharides. So we can see it here properly. So when it comes to polysaccharides, okay, where do we have polysaccharides? Hold on. Yeah. When it comes to polysaccharides, you can see here, you can have homopolysaccharides or heteropolysaccharides. What is the meaning of homopolysaccharides? If you take an example of starch, cellulose, glycogen, or pectin or dextrin, they are made up of same type of sugar, repetitions. For example, with respect to starch, cellulose, glycogen, they're all made up of glucose, right? So they're made up of one sugar which is repeating to n number of times. So this is about glucose. When it comes to heteropolysaccharide, hyaluronic acid and heparin, they are made up of several different types of heteropolysaccharides, like sugar units. So, all right. So let us come back here. When you talk about monosaccharides, right? So monosaccharides can be again divided into 2 important categories. So those 2 important categories are dependent upon the functional group they have. So based on the functional group they are, you can divide them into aldose and ketose. So let me tell you what is aldose. So if we know that CHO is sugar, isn't it? So R is a functional group, okay, that is aldose. So for that R, if we have CHO group, okay, this is called as aldose. Okay. What is ketose? In ketose case, it's quite different. Okay. For example, RC, and then you have a bracket open and then a double bond O. Okay. And then you have another R group. So this is called as ketose. Okay, this is called as ketose functional group. So if you want me to write a structure about it, okay, I'll write a generic structure so that you will understand what is aldose and what is ketose. Okay. So for example, if I have carbon, and carbon. Okay. So for this carbon, I will attach a double bond O and a H group, and then I have OH, and then I have OH. Okay. And then I have H, and then I have H, and then I have H. So this we call it as aldose. Okay. Did you understand? See, you can recognize this structure. So I'll draw a line here. Okay, I'll draw a line here. Sorry if it is tilted. Okay. Let me correct it. Okay, let us try for ketose. What is ketose? I will write 3 carbon atoms. Okay, it is a triose sugar. 3 carbon atoms I have written. So I will write it as H and then OH here. So here what happens? I will draw a double bond O, and then I have a OH here, and then I have a H here, and then I have a H here, and then I have a H here. So you can see the difference between what is aldose and what is ketose. Okay. So based on the functional group, we can divide our monosaccharides or sugars into aldoses and ketoses. Okay. So after we divide them into 2 functional categories, based on the number of carbon atoms that are present in a sugar, we can determine them into several classes. So what are those? So if we have 3 carbon atoms, we call it as trioses. If we have 4, we have tetroses. If we have 5 carbon atom, we call pentoses. If we have 6 carbon atom, we call it as hexoses. Okay. Then if we have 7, it is called as heptoses. Okay. Is it clear? So you can see the examples of aldoses and you can see the examples of ketoses. So some of the examples are glyceraldehyde is an example for aldoses. And then you have dihydroxyacetone. Okay. So dihydroxyacetone is an example for ketoses. So similarly, you have several examples for what are the different types of sugars based on their functional group and based on the repetition of their number of carbon atoms in their structure. So coming to oligosaccharides, as I told you, again, we have disaccharides, trisaccharides, and tetrasaccharides. What is disaccharides? For example, 2 different types of sugars are involved in disaccharides. Trisaccharides are, you have repetition of 3. If you go to tetrasaccharides, you have repetition of 4 different types of sugars. When it comes to polysaccharides, we have homopolysaccharides, as I said, and we have heteropolysaccharides, and we discussed about it, right? So coming to derivatives of sugar, what is sugar derivatives? If you have any other functional group attached to your sugar group, then you call it as derived sugars. So for example, if you have an amino acid attached to your sugar group, that is called as amino sugars. For example, glucosamine and galactosamine are the examples of your amino sugars. For example, deoxy sugars. Okay, when you have a removed group, when the oxygen groups are removed, then you have deoxyribose, and then you have your fucolase. Okay, fucolase is another example. So alcohol derivatives you have acid derivatives and other derivatives of your derived sugars. So what are derived sugars? Whenever you have other functional group like amino acids, removed hydroxyl groups, or when you have alcohol groups attached to your main structure, or you have acidic groups like carboxylic acid attached to your sugar structures, or any other functional groups attached to your sugars, then they function as derived sugars. Okay. So this is about polysaccharides. And one of the important criteria about homopolysaccharides is, as I said, when you want to test a homopolysaccharide, usually you use iodine test. For example, if I take cellulose and sucrose, okay, and then I add iodine to it, we know that when we add iodine to our sucrose, it turns bluish color, right? Whereas cellulose, it does not turn bluish color. Why? Because of their elaborate polysaccharide net structure. If you see cellulose is a linear structure. It will be in a straight line. Whereas when you go for your starch will have something called as helical structure. Okay, a complex helical structure. So because of this helical structure, now what happens is the iodine molecules will be held, okay, within the starch molecule, and that is how we identify whether the starch is present in our food ingredient or not. Now, carbohydrates are very, very important to our body. Why? Because they are very rich energy source. They're ready energy source for our body. Okay. So they are present in many of our staple foods like grains, fruits. Okay. The sweetening we get from our fruits is fructose. Okay. The table sugar that we add for our tea and coffee on our other sweets, it is sucrose. All right. So, and the cell wall is made up of cellulose. So similarly, n number of applications are there when it comes to carbohydrates within the living system. All right. So let us go for one of the other important, biomicromolecules, where we are talking about proteins. Okay. So what are proteins? Proteins are basically the polymers. Okay. We can call it as heteropolymers. Okay. We can call it as heteropolymers. So they are heteropolymers of what? They are heteropolymers of amino acids. All right. So what are amino acids? So when I tell you that it is heteropolymer, that means it is made up of different types of monomers, isn't it? Hetero means different. Polymer means it is made up of subunits or monomers. So I am telling you it is heteropolymer of amino acids. Okay, kids. So when we talk about amino acids are of 20 different types. Okay, there are actually 20 different types of amino acids. I'll just erase this. Give me a second. Yeah, 20 different types of amino acids I can tell. So based on our requirement, okay, based on our requirement to the body, we have essential and non-essential amino acids. So if I say non-essential amino acids, there are around 14 amino acids. Okay, there are 11 amino acids that are non-essential, and there are 9 amino acids that are essential. What are the essential amino acids? Which we have to get it through our food. What is non-essential amino acid? Something which our body can synthesize by itself. Okay. So what is amino acids basically? So amino acids are very, very simple. I will give you a small hint to remember the structure of amino acid. You know the structure of CH4, isn't it? Right? Methane. So let us draw the structure of CH4. Okay. So we can write it like this, isn't it? We can write it like this. So to this structure, what will I do is I will keep the carbon as it is, and I will keep one of the hydrogen as it is. Okay. So let me change this. Okay. Let me change this so that it will be easy for you. Okay. Let me keep this hydrogen, the lower hydrogen as it is. And I will knock out this hydrogen and I'll replace it by R. And then I will knock out this hydrogen and I will replace it with NH2. Okay. And then I will knock out this hydrogen and I will add carboxyl group COO minus or COOH. So this is a general structure of amino acid. So what do we have in an amino acid? We have an N-terminal or amino group, right? We have an amino group, and then we have a carboxyl group, which is an acidic group. Okay, acidic group, and then it is also called as C group or carboxylic group. Okay, and then we have a functional group, and then we have an hydrogen atom. All right. So based on the characteristics of your R group, your amino acid nature also change. It can be acidic, it can be basic, it can be neutral, it can be polar, it can be non-polar, etc., etc. Right? It can be positively charged amino acid, it can be negatively charged amino acid. Correct, no people? So based on what functional group we add, our amino acid characteristics also change. So if we look at the chemical nature of our amino acid, what we can find is, since our amino acid exhibits, okay, positive charge in the N-terminal and negative charge in the C-terminal. You can see here in this picture, there is positive charge here and there is negative charge here. Okay. So since they have both positive and negative charge within the same chemical structure, we call these ions as Zwitter ionic forms, or Zwitter ion we call it as. So our amino acids are Zwitter ions. So because of the characteristics of Zwitter ion, now what happens is, the amino acids will start making, okay, polymers. So how do they make polymers? So if I take two different amino acids, okay, if I take two different amino acids, okay, let us take amino acid 1, okay, and amino acid 2. Now, how does this amino acid 1 and amino acid 2 bind to each other? So the N-terminal of one amino acid will bind to the C-terminal of another amino acid through a peptide linkage. Okay. So that is a covalent bond. Okay. During this process, what happens, So during this process, a water molecule is removed. Okay. So then you have one amino acid and the second amino acid combining with each other. So like that, you have n number of different amino acids combining together to form a chain called as polypeptide. Okay. Polypeptide chain. So these polypeptide chain, it can be one, it can be more than one, or it can be 2 or 3 different polypeptide chains. They combine together and then they become proteins. Okay. So now proteins, when it comes to our biological system, they perform around 98% of all the biological functions that we know about. So they're pretty important. Whether you take about collagen, which is present as a ground substance within your intracellular spaces. Okay, if you talk about trypsin, which acts like an enzyme, insulin, which acts like an hormone, antibodies to fight our infections, okay, to give your chemical signals from one cell to another cells and cell to cell communications, okay, your reception, most of the receptions that happens, and to even transport your glucose, okay, amino acids are, proteins are very, very essential. So proteins are structural molecules, all right? So they're highly structural molecules. So these structures determine the function of protein. Whenever there is a denaturation in the protein or the structure of protein is destroyed, then the protein becomes pretty useless and it will not perform any function. So here is an example of some of your amino acids. Okay, you have glycine, alanine, and serine. You can see here H, CH3, and CH2OH, which are the functional groups. Okay. So we talked about protein and protein configuration. So basically proteins can be classified based on their structure. You can have primary proteins, you can have secondary proteins, you can have tertiary proteins, and you can have quaternary proteins or quat proteins. So these primary protein, secondary protein, tertiary or quaternary protein structures depends upon the complexity of protein structures. If I'll just quickly summarize this, when it comes to primary structures of protein, it is a single polynucleotide chain. Okay, so where you have only one polynucleotide chain. When it comes to secondary proteins, it is made up of two different chains, where you have one type of peptide chain which is called as we can call it as beta pleated sheet. Okay, beta pleated sheet, and then you have plus, you have one more type of peptide chain called as alpha helix. Okay, so these two different structures of your protein chains combine together to form secondary protein. When it comes to tertiary proteins, you have one single polypeptide chain, but what happens based on the type of amino acids that are present. Now, for example, there is positive, let us say that there is positively charged amino acid here, and there is negatively charged amino acid here. So now what happens? Positive, negative will attract each other and they will form a bond, isn't it? So you can see ionic bond, you can see hydrogen bonds, you can see disulfide bridges. So what happens? This linear polypeptide chain, it will fold in three-dimensional space to form a tertiary structure. Now this is called as tertiary proteins. When it comes to quaternary proteins, the quaternary proteins are basically combination of several different polypeptide chains. Okay, they are very complex proteins. For example, hemoglobin. If I take hemoglobin, it is called as a metalloprotein. You know that it is in your red blood cells. So there is iron atom. So Fe2 plus L, right? And there are four chains. Okay, four different types of polypeptide chains. Two are called as alpha alpha and other called as beta beta. You can see four different polypeptide chains combined together in one three-dimensional space to give rise to more complex protein structure that is called as quaternary structures. Okay, I hope you understood proteins. And if you have any doubts, you can just put in a chat box so we can try to solve the doubts. Okay, people. Next one, we'll go for lipids. Okay, what are lipids? Lipids are basically hydrocarbons, okay, which are combined to a glycerol molecule or an alcohol group, we can call it as. For example, there are two important ingredients. Okay, lipids are of many types. We can call it as fats or oils. Okay, and then you have your derived lipids like sterols, you have waxes, okay, etc. So these are some of the examples of your lipids. So if I take fats and oils, okay, so if I take an example of fats, okay, let me solve about fats. Okay, so fats, when do we have a fat? So fats can be of three types. Okay, it can be monoglycerides or diglycerides or triglycerides, okay, based on the structure. So let us break it down. First of all, for a fat molecule to form, we have two ingredients. What are the two ingredients? First one we have is a glycerol, and the second one we have is a fatty acid chain. Okay, so let us understand what is glycerol and what is fatty acid. Glycerol is basically you have three carbon atoms, okay, and then so basically you can write it like this: CH2, CH, and then CH2, and then you have OH, and then you have OH, and then you have OH. So this is called as a glycerol. So in a glycerol molecule, you have three carbon atom, and then you have 5 hydrogen atom, and then you have 3 OH groups. Okay, this is called as a glycerol. To this glycerol, what happens, so you need a glycerol first, and then what happens, you have a fatty acid. So what is fatty acid? Fatty acid is basically a long chain of hydrocarbons. Hydrogen and carbon chain will be there. You can see here saturated. So you can see here carbon, 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 right? There is hydrogen, hydrogen, and hydrogen. At one end of this chain, there is COOH group or acidic group or carboxylic group attached to it. So this is called as a fatty acid. Okay. So now for this glycerol, okay, if you have your fatty acid chain attached, okay, a three different position. Okay, this is also fatty acid, this is also fatty acid, this is also fatty acid. Okay, sorry. Okay, this is also fatty acid. So since we have three fatty acid chain, isn't it? So now we call it as triglycerides. Okay, triglycerides. Sorry. Okay, triglycerides. So triglycerides are nothing but your fats. Okay, so now we understood. So that for the fat molecule to form, we need one is a glycerol, second one we have is a fatty acid. So when they combine together, we get something called as fats. So here it is given. Okay, glyceraldehyde or triglycerides, what we call it as. Okay, so we have, see, R2, R1 and R3 represent what? Fatty acid chains. And the C are there. No, this will represent what? Our glycerol molecule. All right. So based on the type of fatty acid chain that is attached to our structure, we can have two different types of fatty acids or fats. Basically, we have saturated fats and unsaturated fats. What are saturated fats? If you see the saturated fat structure, people, so you can find only single bonds, isn't it? You can see only carbon single bonds. So when they have only single bonds in a fatty acid chain, that means you cannot add any more hydrogen atoms to it. When you cannot add any more hydrogen atoms to it, that means it is saturated, right? So these are called as saturated fats. When it comes to unsaturated fat, you can see here in the structure, in the chemical structure, you have a double bond here. So whenever there is a double bond, that gives you a scope to add more hydrogen atom to it. That is called as unsaturated fat. So based on the position of your double bonds, you can again divide them into two different types. What are the two different types of your fats? You have cis fat and you have trans fat, isn't it? So unsaturated cis fat, I will write it here. Okay, you have cis fats and then you have trans fat, right? So what is cis fats? Cis fats are the structures where you have double bonds. For example, if you have a chain of your fatty acids, and if the double bonds are present on the same geometric position, that is called as cis fats. What is trans fat? If you have your structure and if it is present on different geometric positions, it is called as trans fat. Okay, so usually cis fats are quite good for your body. Basically, they reduce your cholesterol level and the plaque levels in your body. When it comes to trans fat are artificially made. So basically, what do? You take a saturated fat and you pump hydrogen. Okay, when you hydrogenate it, now double bonds will be formed between your structure. So that will give rise to trans fat. For example, vanaspati ghee. So one of the characteristics of your trans fat is they will solidify very fastly when it comes to colder climates. So you can see your coconut oil or your ghee, it will immediately solidify with respect to lower temperatures, isn't it? So that is the problem with trans fats, and trans fats are very unhealthy. So basically they will form a lot of cholesterol deposition in your body. Okay, unwanted lipid deposition will take place in your blood vessels, and that might lead to cardiovascular problems. Okay. So you can see here lipids can be divided into two. It can be divided into fatty acids and it can be divided into glycerols. Okay, we understood what is saturated and unsaturated. Okay, so now our important focus is to understand about phospholipids or phosphoglycerides. So what is phospholipids or phosphoglycerides and why it is important? So phospholipids or phosphoglycerides are very, very important because they make up the important composition of our plasma membrane, isn't it? So our plasma membrane is made up of phospholipid, right? So that is why we have to study about phospholipids. So what is the composition of phospholipids? So first we have a glycerol molecule. All right. To this glycerol molecule, now what happens? One saturated and one unsaturated fatty acid chains will be attached. Okay, the second one is done. First we have a glycerol. To the glycerol, what do we have? We have a fatty acid molecule that is attached to it. Next one we have is your phosphate group that is attached to the third carbon atom of your glycerol molecule. Now, phospholipids are very, very unique. Why they are very, very unique is because when you take your fats, waxes, or your cholesterol, they are all hydrophobic in nature. That means whenever you put in water, they float in water, they do not dissolve. But when it comes to your phospholipids, they are amphipathic molecules. What is amphipathic, people? Amphipathic means they behave like amphibians, right? That means they are both water loving and water hating, right? So when you see your phosphate group that is present in your phospholipids, they interact with you. The phosphate head is there, no, that will interact with your water very well. That is called as hydrophilic end or hydrophilic head, we will call it as. Whereas the long chain of fatty acid is there, right? So they are hydrophobic in nature. They are water repelling or water hating in nature. So whenever we put phospholipids in water, they will form micelle structure. Okay, so that means all your hydrophobic heads will be pointing towards outside where they will interact with water, and all your hydrophobic tails, okay, will be within and it will be highly protected. So because of this characteristic nature of your phospholipids, right, that is how your plasma membrane is able to create this semi-permeable membrane, which can protect the entire composition of cell from not being destroyed because of the water constituent in your body. Okay, so next category of your lipids is sterols, and one of the best way to remember sterols is they have ring structures. So basically there are 4 rings to the cholesterol molecule, and there is one OH molecule attached to this cholesterol ring. Okay, so these sterols are basically very essential to your body because of the news and all this propaganda. We think that sterols are bad. Cholesterol is very essential because they are important for the synthesis of certain growth hormones and sex hormones like testosterone, progesterone, okay, etc. So this is about your lipids, and the next we are going for is nucleic acids. Okay, so people, when it comes to nucleic acids are basically your DNA and RNA are your nucleic acids. So they are also heteropolymers. Okay, heteropolymers of what? They are the heteropolymers of something called as nucleotides. Okay, nucleotides. So when I talk about nucleotides, they are made up of 3 important ingredients. Okay, we are talking about nucleotides, people. So when we talk about nucleotides, they are made up of 3 ingredients. What are those? One is nitrogenous bases. Okay, I'll write it here. Sorry, nitrogen bases. And the second ingredient is your sugar molecule. Okay, sugar molecule. And the third ingredient for it is a phosphate group. Okay, so these 3 combine together, okay, to form what? A nucleotide. All right. So what are nitrogenous bases? Nitrogenous bases are of 5 types basically. One is adenine, and then you have uracil, thymine. Okay, where is it? Okay, we have, wait, let me write it here. We have adenine, thymine, cytosine, guanine, and uracil. These are the 5 nucleotides that are present. When it comes to DNA, it is ATCG, that is adenine, thymine, cytosine, and guanine. When it comes to your RNA, it is AUCG, adenine, uracil, guanine, and cytosine. So instead of T, there is U in RNA. So when it comes to your nucleotides, basically, it is made up of sugar, I told. And the type of sugar that is present in your nucleotide, it is called as pentose sugar. Okay, it is called as what? Pentose sugar. So we are talking about this, no, sugar groups. So this pentose sugar has what? 5 carbon atoms. Okay. When it comes to DNA, it is called as deoxyribose sugar. When it comes to RNA, it is called as ribose sugar. Okay. So with these 5 carbon atoms, now what happens to this sugar? Your nitrogen base and phosphate group attach together to form a nucleotide. For example, you can take this structure. All right. So you have your sugar group, right? So to this sugar group, you can see here, this is the first carbon atom, this is the second carbon atom, this is the third carbon atom, this is the fourth carbon atom, this is the fifth carbon atom. Okay. So to this, for the first carbon atom, what happens? A nitrogen base will attach. Okay, your adenine, thymine, cytosine, or guanine or uracil will attach. Through what linkage? Through N-glycosidic linkage. All right. Now to the fifth carbon atom, what will happen? A phosphate group will attach. Okay, through what linkage? Phospho ester linkage. Okay, so when you have a sugar group where you have a nitrogen base attached to your first carbon atom, and then you have your phosphate group attached to your fifth carbon atom, in combination it will form one nucleotide. Okay, so when it comes to nitrogen bases, people, we can divide them into 2 important categories. What are those? One class of nitrogen bases. So I told you these are the nitrogen bases, isn't it? So, right? So if I take adenine and guanine, okay, A and G as two nitrogen bases, and cytosine, thymine, and uracil as another group of nitrogen bases, now we can categorize them into two important categories based on their structure. So adenine and guanine belong to a category called as purines. Okay. Whereas cytosine, thymine, and uracil, they belong to a category called as pyrimidines. Okay. So what is purine and pyrimidine? If you see purines, they are made up of heterocyclic structure. You can see here. One ring is made up of 6 atoms, one ring is made up of 5 atoms. Okay. So one ring which is made up of 6 atoms is called as purine ring. And one ring which is made up of 5 carbon atom, it is called as imidazole. Okay, imidazole ring. All right. So since it contains two different types of rings, then we call it as purines. When it comes to pyrimidines has only one ring, and the name of the ring itself is called as pyrimidine ring. That is why cytosine, thymine, and uracil are called as pyrimidines. Okay. So this is about nucleotides. Okay, people, too much talking, but now we have to solve a question. Okay, so here is a question: which of the following have carbohydrate as prosthetic group? Okay, I'll launch the poll for now. Okay, you can try to answer this question. Okay, I hope you can see. Okay, can you see? Yeah. Oh, come on. Oh, sorry, people. What happened? Where's the polls? Sorry. Okay. You have answered C. Only one have answered C, and I can see we have a lot of audience today. So, can you see the polls, people? Yeah, 2 have given C. Come on, people. 3, 10 seconds. 4 on 4. Okay. Okay. Time up. I'll try to end the poll. Okay. So let us come back to the question. Which of the following have carbohydrate as a prosthetic group? I believe. Okay, let me summarize again. Your answer is C, right? So you're telling lipoprotein, no? So all of you have answered lipoprotein as a prosthetic group, people. So you can see here, lipo is lipid, isn't it? Protein is protein, right? Where is carbohydrate here, right? Carbohydrates are sugar, isn't it? So where can we find sugar? Glyco, right? Glyco represents sugar unit. And then you have protein, right? So carbohydrate is involved as a prosthetic group in what? Glycoprotein, isn't it? So observe the question. Your question itself has an answer. Okay. Here only it is carbohydrate. So it's as simple as that. All you have to do is glyco. Search for glucose. Okay. So your answer will be right. So anyways, no problem. We can have more interaction and then we can get our answers as right as possible as we move on. Okay, fine, people. So we have the final part of our discussion where we have to talk about enzymes. So we know that what are enzymes? We have heard about enzymes a lot in our lower classes as well as in our daily life when we talk about it, right? What are enzymes? Enzymes are biocatalysts, okay, which are essentially proteins, okay. I can tell you when it comes to enzymes, all enzymes are protein, but all proteins are not enzymes. Okay, remember this statement. Okay, all enzymes are proteins, but all proteins are not enzymes. This is very, very essential for you. So usually when the enzymes are formed, they are in a colloidal state. Okay, they are often produced when they are produced in an inactive state. They are not active. Immediately when the enzyme is produced, they cannot be active. Why? Because many of these enzymes are digestive enzymes, and some of them convert, some of them isomerize, some of them will transfer certain prosthetic groups here and there. Okay, they're highly reactive molecules. So first of all, our question is, what are the different types of enzymes that we can come across? So we have basically 6 different types of enzymes in our biological systems. What is that? First, we have oxidoreductase, transferase, hydrolases, lyases, isomerases, and ligases. So each of them, see, these are the classes of enzymes. So within this, there are like several enzymes that are, placed into them. For example, if I tell oxidoreductase, the name itself says it oxidizes and it reduces, right? For example, it's given here, right? It acts as an electron acceptor in the case of NAD plus in the dehydrogenases, right? With the help of dehydrogenases. Similarly, it oxidizes with respect to H2O2, that is peroxidases, where you have a hydrogen peroxide, and with the application of peroxidases, now what happened? It gets oxidized, right? Now, when it comes to transferases, what is transferases? You have certain prosthetic groups or functional group like amino group, phosphate groups, or carboxyl group, or carbonyl groups. Now, what happens? Two different functional groups will get transferred between them. So this is called as transferases. Ketases, okay, transaminases, phosphorylases are the example for transferases. When it comes to hydrolases, so what it does, hydro means what? Hydrate. So it adds water group, right? So basically it adds water group and it helps in breaking down of some of the important covalent bonds like phosphodiester bond. Okay, so when it is broken and it does this function, this is called as hydrolases. What is lyases? Lyases, what happens? For example, the groups like carbon dioxide, ammonia are either added or removed from the entire structure of a particular compound. So this is called as lyases. What is isomerases? We have heard about geometrical isomerism, right? Cis isomers, trans isomers, and then their rotations and all. So if they are able to change their from cis to trans or trans to cis, so this function is done by isomerases. What is ligases? Ligases are means stitching, okay? So basically what they do is they take two different molecules and they try to make new bonds between them and then make the structure bigger. So this is called as ligases. So this is a classification of enzyme based on their functions. Now, when it comes to the energy, so usually in our biological system, what happens? There is a tremendous demand for the energy when it comes to our metabolic functions. So if we want to break down certain things to make simpler compounds, for example, we eat complex food, it has to be broken down into simpler compounds, or you have to signal something, or you have to convert something. Now, breaking of bonds require energy, isn't it? So initially there is an input of energy that is required. Or if we want to convert a substrate into some product, you have to apply some energy. So this requirement of energy has to be reduced as much as possible in our biological system so that we have, net. For example, after the consumption of food, after the utilization of food, and after everything, we should be energized. We should not go in low energy, right? So to decrease the energy demands, enzymes will help us. So you can see in this graph, now what happens without the application of energy, look at the amount of activation energy that is required to convert substrate into product. Whereas with the application of enzyme, now what happens? Activation energy that is required is quite less. Now, how does it happen? Now, initially what happens is to the substrate, an enzyme reacts and forms substrate enzyme complex. And this complex is an intermediate product, okay? So this intermediate product to convert itself into product and enzyme, okay, product and enzyme derivatives, it requires very low energy. So this intermediate transformation between substrate and substrate enzyme complex and finally product enzyme resultant, okay? So this reduction of the energy is done by what? It is done by your enzyme. So this is how it usually happens, okay? So when it comes to enzymes, enzymes are highly specific to their substrate. They do not simply go and act on any random substance and then try to convert them, all right? So they're highly specific in their site of action or the substrate of action. So that is what one of the key criteria of your enzymatic reaction. And one of the important mode of actions of enzymes is the lock and key mechanism. What is lock and key mechanism? We know that when you have an enzyme and when you have a key, when you have a lock, sorry, if you want to open the lock, you require an appropriate key. So you cannot just simply use any other key and open the locks in all the cases, right? I'm mentioning all the cases because I know you people, you might think, oh, we have universal key and all that things. But just imagine an unopenable lock and can be opened by only one key. So that is how enzymes work. So substrates, usually what happens is substrates have certain sites on their structure where they provide binding site for their enzymes or enzyme substrate binding sites. That means what? They can easily come and sit at a particular position and they can cause this enzyme substrate complex. Only when they cause this enzyme substrate complex because of this lock and key mechanism, okay, then the catalyzation of this reaction happens where the conversion immediately transforms to enzyme product complex. And finally, when the enzyme product complex forms, no, now the affinity between these two will reduce. Initially, what happens? These two attracted very well. These two formed the bond very fastly. After their complex, after the catalysis reaction happened, now what happens? The affinity of this product with this particular enzyme decreases. Since the affinity decreases, now what happens? The enzyme will depart and the product will also depart. Okay, that is how the enzyme is able to come out of this enzyme product complex. Okay, because of the decreased affinity between enzyme and product, whereas there is high affinity between enzyme and substrate. Okay, so this is about it. Now when it comes to enzymatic reactions, have you ever thought about why your body temperature is always same, why your pH is always balanced in your body, why you can to go highly acidic or highly basic? It is because all our biological reactions are mostly affected because of the enzymes, and for an enzyme to have the highest rate of action, they require very appropriate or optimum temperature, optimum pH. Okay, a very perfect substrate is required. The enzyme itself is required in a proper quantities or volume for the enzymatic reaction to take place. Okay, so inhibitors should not be there. If the inhibitors are there, enzymatic reactions actually decrease. Okay, so what type of end products do we have? Okay, do they inhibit the rate of reaction or they increase the rate of reaction? Okay, are there any activators which support the enzymes for the enzymatic reactions? Usually radiations, presence of radiation decrease the enzymatic reaction. Okay, so some of the examples are given here, very important examples. One is temperature, other one is pH. Look at this temperature. Okay, people, enzymes are proteins, as I said. Usually when the temperature goes down and down, okay, when it completely decreases from the optimum temperature, usually what happens is the protein structure remains intact. Remember this. And the protein or the enzyme becomes what? They will not act. Okay, they will not function. Okay, they will not denaturate, but the function of this enzyme is decreased. All right, as the temperature increases, now when it reaches the optimum temperature, the enzymatic reaction is at the highest. Okay, they can give you the highest enzymatic product. Okay, as the temperature goes higher and higher, because of its protein structure, okay, enzymes are proteins, isn't it? Now what happens? There is a possibility of this structure getting denaturated. The bonds will break, amino acids will get distorted. When the structure of an enzyme goes, okay, is destroyed, then we call it as denaturation of enzyme. When the denaturation happens, the enzyme becomes pretty useless. Okay, similarly with respect to pH, only during the optimum pH you can see there is highest, okay, there is highest enzymatic activity. So you can observe here what is the pH. Usually the 7.5 to 7.8 is the ideal pH for most of the enzymatic reactions that happen in our body. Okay, when as it goes to acidic, now the enzymatic reactions decrease. As it goes more alkaline, the enzymatic reactions will also decrease. So this is about the pH. Now coming to substrate concentration. What is substrate concentration? When you have enzymes, enzymes should actually react with substrate, isn't it? If I have 100 enzymes, okay, if I have 80 substrate, okay, can you now imagine here the amount of enzyme is quite high, correct? No, for example, I will give you one more example. If I have 100 enzyme molecules and if I have 1000 substrate molecules, now what happens, people? The enzymes, all these 100 will occupy this substrate and 900 of the substrate molecules are remaining, right? Correct? No, now can you expect more enzymatic product? No. So the substrate to enzyme concentration is very, very essential. Okay, so now what happens with the increase in substrate concentration? At one point, what happens as the substrate becomes more and more, all the enzyme molecules will be involved, okay, will be totally occupied for this enzymatic reaction, and finally it reaches a point of saturation where you can where you cannot expect more product out of this chemical reaction. Okay, so this is about enzyme substrate concentration. Okay, so basically what happens as you increase the concentration of substrate, the enzymatic reaction also keep on increasing, and it will reach to the maximum velocity or maximum output. After the enzyme molecules are completely occupied and you don't have any enzymatic concentration, it will reach saturation point where you do not where you cannot expect more and more product. So when it comes to enzymatic reactions, basically enzymatic reactions are affected by the presence of inhibitors. So inhibitors are molecules that are similar to that of your products. Okay, so they occupy these active sites of the enzyme and they do not allow your substrate to come and bind to their active sites. Okay, so you can see that there are two different types of inhibitors. You have competitive inhibitors and you have non-competitive inhibitors. Competitive inhibitors are very vigorous. Why? Because they have almost similar structure to that of your substrate. Because of that, now what happens, they occupy these active sites before the substrate can occupy, and they have high affinity towards this enzyme than the substrate. So now your substrate cannot bind to this active site and the enzymatic reaction cannot take place. When it comes to non-competitive inhibitors, now what happens, they do not bind to these active sites on the enzymes, they bind to this another different site in the enzyme. Now the enzymatic configuration only will change, the structure only will change. That will result in what, the entire substrate binding reaction is blocked. So because of that, you don't have the result or the outcome of this particular enzymatic reaction or inhibition. Okay, so you have the example for competitive inhibition. For example, in case of succinic dehydrogenase, which is an enzyme, okay, instead of succinic dehydrogenase, if I add, okay, malonate, which is similar structure, okay, to that of succinate, now what happens, it will go and block its sites. That will avoid it to form any of the enzymatic reactions. All right, so finally we have to talk about Michaelis-Mendelian Michaelis-Menten equation or constant. Okay, but we are basically coming to the end of the session. We have 3 minutes. Okay, so we have homework here, people. Okay, homeworks are there and there are certain questions. Okay, so finally you take this as a homework. So next session when we come, I will talk a little bit about the rest of the topics that are remaining so that we can talk without any problem. So if you have any doubts, please let me know and I will launch the polls for NPS. Okay, please submit it as soon as possible. Can you see the form, people? Is the NPS form visible? Okay, I think it should be there now. Please do it within a minute so that if you have any doubts, we can still. Students, please try to fill up this NPS form. Is it visible? Yes. You have 5 questions. Okay. Only one have answered. Can others give your reply, please? It is very essential to keep the class quality. Okay. So your feedback would be very helpful so that to improvise classes in the coming sessions. It's fantastic. We have people from so many different parts. Okay. Pune, I can see so many different places. Kolhapur. It's amazing. Yeah, students, you know what? If you have any doubts or any problems with the class, you can just put it in the chat box, okay? Or when you come to the next class, you can just list down your topics of doubts and then become competitive because you have weekly classes too. So when you GEAT examination, if you face it very properly and you compete very well and you get better marks, so you'll be indulged in the weekday classes too. Okay, anyways, the videos are provided for you in the weekday sessions too. But when you are in the class, I think it will be more interesting for you and you will understand even better because of the elaborate lectures. So take this examination seriously and try to focus on this session, on the particular exam so that you can involve in daily sessions. Okay. So thank you very much for your response and thank you for the wonderful class today. Okay. I was happy to have such audience. Okay. See you. I'll see you in the next week. Okay. For the coming classes. Thank you very much.